Hi students, welcome to the online lecture series of Vignans University. My name is Abraham Karlapudi. I am an assistant professor of biotechnology in Vignans University for Science, Foundation, Technology and Research, GM2B University. Today's topic is tertiary level of protein structure. The tertiary and quaternary structure actually looks quite similar. They both describe the arrangement of a protein, the three-dimensional arrangement of a protein, the polypeptide chain which turns the three-dimensional arrangement in its local environment at a higher level than a simple secondary structure. And the tertiary structure is the arrangement of alpha helices, beta sheets, loops, turns within the single polypeptide chain within the natural local environment. There are three-dimensional arrangements, whereas the quaternary structure has rearrangement of several polypeptides of this tertiary structure. Those polypeptides form into a protein complex and that performs a specific cellular function. The tertiary structure is built from motifs to domains to proteins. Motifs are structural units and domains are functional units. These together to form a protein. You can see the alcohol dehydrogenase as a cartoon representation with helices in green and sheets in blue and loops in grey. The smallest grouping of secondary structure is called a motif. That means a motif is made up of several structural elements. That means structural elements are nothing but alpha helices, beta sheets. These are assembled in a consistent way to form a motif. Motifs recur in many different proteins with varying sequences. A very common motif which we can see here is beta alpha beta motif, where two adjacent beta strands are connected by an alpha helix, and see how this allows the formation of a beta parallel sheet. One or more motifs can assemble to form a compact globular structure that is called a domain. A domain is a functional unit with a compact globular structure. Here you can see the C-terminal beta alpha beta motif and highlighted in yellow color. This is the one which is interacting with the additional beta strands and several other helices to form a functional domain. These domains are independent folding units within the proteins and can usually fold on its own and maintain its tertiary structure independent of the rest of the protein. Proteins often has multiple domains. These may interact with each other to form a functional unit. You can see in this example of the protein, the domain is seen in yellow interacting with a second domain which features a Rossmann fold. It is nothing but a Rossmann fold. The Rossmann fold is a tertiary fold which is found in proteins that binds to nucleotides such as enzyme cofactors. These enzyme cofactors are FAD, NAD+, and NADP+. This Rossmann fold is composed of alternating beta strands and alpha helical segments, where the beta strands are hydrogen bonded to each other, thereby forming an extended beta sheet, and the alpha helices surround the both faces of the sheet to form a three-layered sandwich. These sites of enzymes are often found in loop regions. The tertiary structures is having some important principles. The secondary structures form wherever it is possible. Where the environment favors the secondary structures to form into a motif for a domain, the secondary structures favors to form a possible structure. That means due to the formation of a large number of hydrogen bondings and hydrogen bonds, the secondary structure forms wherever it is possible. And these helices and sheets often pack close together by forming the backbone, and the backbone links between the elements of secondary structure which are usually short and direct. And the proteins take a fold, and the proteins folds to make the most stable structures by making the hydrogen bonds and to minimize the solvent contact. Two general classes of proteins based on the tertiary structure. The main important classification of proteins are globular proteins and fibrous proteins. Globular proteins are folded into compact, spherical and globular shape and also soluble. There are several types of secondary structures which you can find in the globular structures. The function of the globular structures by maintaining the various cellular functions, the mostly enzymes and globular proteins. Here you can see the example of carbonic anhydrase. The next is fibrous proteins. Fibrous proteins are long strand fibrous proteins with elongated, tough and insoluble. It has the insoluble nature. The single type of secondary structure which you found in the fibrous proteins is sheets. Function of fibrous proteins: they have different functions. They play a good role in structures, forming the structures and giving the support and the shape and external protection. Here you can see the example of collagen. Forces that stabilize all globular proteins. Globular proteins are compact. The polypeptide chains are folded into spherical or globular shape. The peptide chain that must satisfy the constraints inherent in its own structure. That means the peptide chain must fold so as to bury its hydrophobic side chains inside and minimizing the contact with the water. The peptide chains are composed of L amino acids, which has a tendency to undergo a right-handed twist. There is no space inside because the water is effectively excluded from the hydrophobic interior region of the protein. Nearly all buried hydrogen bond donors, example serine, form bonds with hydrogen bond acceptors, example glycine. The bond formation neutralizes the polarity of hydrogen bond. Well, the tertiary structure refers to the spatial arrangement of amino acids in the three-dimensional space. That means they are found far away from one another along the polypeptide chain. There are several important interactions that are involved while forming the tertiary structure of the protein. There are few interactions. Out of those interactions, the one main interaction is the hydrophobic interactions. Most proteins exist in aqueous solution. We know that when non-polar molecules are placed in water, they will aggregate together because this will create a thermodynamically more stable system. What does that tell us about the way in which the polypeptide will fold? The amino acids with hydrophobic side chains, example valine, leucine, etc., will tend to be found inside the protein. Those amino acids that contain hydrophilic side chains, example lysine, aspartate, will tend to found on the outside of the protein, on the surface of the protein. A protein in an aqueous environment will have an hydrophobic as well as hydrophilic surface. Next is van der Waals interactions. This is one of the most important interactions next to the hydrophobic interactions while maintaining the tertiary structure of the protein. The non-polar amino acids of the protein core interact with one another with an instantaneous dipole moments. Although these van der Waals forces are relatively weak on an individual base, but the aggregate effect of these interactions with many non-polar amino acids create a substantial binding effect, you can see. The next one is disulfide bridges. In some proteins, usually the ones that is destined to be extracellular, the polypeptide chains can be cross-linked via disulfide bonds between the cysteine residues. The cross-linked units are called cysteines, and you can see the reaction of oxidation to form a disulfide bond between the two cysteine residues. The thiol groups are involved. Next is the hydrogen bonds. Hydrogen bonds, the polar and hydrophilic side chains on the surface interact with the water molecules via these hydrogen bonds. These are one of the most important bonds while forming a tertiary structure of the protein. And next one is ionic interactions. Two oppositely charged side chains, charged amino acids, can interact via ionic bonds. For instance, lysine can form an ionic bond with an aspartate. The next coming to the protein solvent interactions. The hydrophilic amino acids, aspartate, glutamate, lysine and arginine, like these amino acids tend to interact extensively with the solvent in context of the folded protein. The interaction is mostly ionic and favors the hydrogen bond. There are instances of hydrophilic residues being buried in the interior of the protein. Often, the pairs of these residues form the salt bridges. And the hydrophobic amino acids like isoleucine, leucine, valine, these tend to form the core of the protein. That is, they are buried within the folded protein. These some hydrophobic residues can be entirely exposed or partially exposed. There are some neutral amino acids like glycine, alanine. They are less preference for being the solvent exposed or not being exposed. You can see the water is interacting with the protein surfaces. Most waters visible in crystal structures make hydrogen bonds to each other, and they act as both hydrogen bond acceptor and donor. Water is not just surrounding the protein, it is interacting with it. You can see the change in the protein structure upon binding of its substrate. The left side is the unbound HIV-1 protease. That means there is a loose conformation. And you can see the right side, the HIV-1 protease bound by a ligand. When bound ligand fits into the active site of the protein, it becomes the thermodynamically stable conformation, and the protein changes its conformation from earlier one to the next one. You can see the first protein structure that was solved by John Kindrew, that is myoglobin, in 1958. This protein was purified from the whale blood. The secondary structure elements, alpha helical fold, is covering the 75% of the total protein structure, and the number of amino acids constitute the myoglobin is 155, and the size is similar to 17 kilodaltons. This is not a simple structure like a DNA. It was solved by John Kindrew in 1958 by using the different techniques extracrystallography and NMR. Moving to the next topic, methods to know the tertiary structure of protein. We already discussed about the three-dimensional structure of protein. It gives you the atomic coordinates. So the tertiary structures are the 3D structures which can give the exact location of each atom in a specific residue and in a protein. The x, y and z coordinates in a Cartesian coordinate system. Then the coordinate structures, they provide the combination of secondary as well as tertiary structures having different subunits. Finally, these give the tertiary structures. There is various experimental methods like X-ray crystallography is one of the finest methods to elucidate the protein crystal structure, and NMR spectroscopy and cryo-electron microscopy. The different methods helps to determine the 3D structure of protein. Well, coming to the X-ray crystallography. X-ray crystallography is a technique to identify the structure of protein crystal. If we have an unknown structure, then crystallize the protein. By crystallizing the protein, you can know the 3-dimensional structure, which can help us to understand further mechanism of action. The size of the molecule is far too small to see with our eye or with our light microscope, because the wavelength of the light is much larger than the size of the tiny molecule. So we have to use the radiation that is much smaller than the light, nothing but X-rays. Each crystal produces millions of protein molecules of same type and arranged in an ordered grid-like structure. By firing the X-rays at these crystals, one can measure the scatter. You can look at the molecular structure of nearly any crystal sample. It takes a long time to grow a crystal. Before crystallization, we need to produce enough protein using genetic engineering cloning principles. X-ray diffraction works based on diffraction grating. The source of radiation is coherent. Same frequency and wavelength are in same phase, and when the atom absorbs and emits the same wavelength, interatomic spacing is of order of an angstroms. Typical distance between atoms inside the crystalline solids is order of angstroms. You can give this equation d sin theta is equal to n lambda, that is the maxima at angle theta. We have to consider an electromagnetic wave which can match the distance and order of 1 Armstrong unit. When an individual atom is under the influence of external oscillating electric field, we have an atom under the influence of X-ray electromagnetic wave. If the atom is under the external oscillating electric field, both the nucleus and electrons are going to experience the same field. Nucleus is much heavier than electrons. Acceleration of the electron which experience the external force. In response, they feel little disturbance and so simple harmonic motion. Electron is bound to the nucleus by simple harmonic motion. Electrons oscillate at same frequency as X-ray oscillates. When electron oscillates, it emits electromagnetic radiation with the same frequency at which the electron oscillates. The electron radiates at the frequency nu as X-ray frequency. If we consider, we have to calculate the intensity of radiation scattered out and reflected theta angle. The wave that is reflected from the first plane is going to be a phase difference with the wave reflected at second plane. And again, the phase difference with respect to third plane that continues. This is nothing but extra path difference. We will have a set of waves, but this wave's phase difference corresponds to the path difference. This gives me the formula 2d sin theta is equal to n lambda. Therefore, n is equal to 2d sin theta by lambda. This is nothing but a path difference between two consecutive waves reflecting from two consecutive planes. If this is the integer multiple of wavelength, then 2d sin theta is equal to m lambda. N is represented by m. M is nothing but a maxima. This condition is for maxima. You can place the protein, the sample, in the center of the instrument and illuminate with a beam of high powerful X-rays using X-ray tube, Coolidge tube. The detector moves in synchronized the motion and the signal is recorded and graphed. You can see in the graph. Well, coming to the nuclear magnetic resonance spectroscopy. The nuclear magnetic resonance spectroscopy is one such technique to elucidate the three-dimensional structure of a protein. In this technique, we will be using electromagnetic radiation in the form of laser. By using this laser, we can illuminate the sample. There is always a good relationship between magnetic field and electric field, and both are perpendicular to each other. The end magnets, which contains the atoms that contains electrons, and the north pole induces these electrons present in the atoms of conductor, and negative charge develops and it creates a magnetic field. This is nothing but induced magnetic field, and if this induced magnetic field affected by the magnet, it can turn the sample to south pole. The transverse magnetic movements producing at resonant frequency of local protons produce a radio signal of the same frequency that can be detected by a coil or a wire. As energy is removed, the protons first move apart in T2 or spin-spin relaxation, and destroy the transverse magnetization, that is followed by T1 relaxation, fall into the lower energy state. That high energy state fall into low energy state when comes from T2 to T1 state and restore the longitudinal movement. We are supposing to provide a 1 Tesla magnetic field, assuming that spin of the proton precises at Larmor frequency. The 1 Tesla is equals to 42.58 megahertz. If this transition radio frequency is exactly matching 42.58 megahertz in the vicinity of the protons, two things can happen. The net magnetization after opposing forces cancelled out. In addition, the sinusoidal radio frequency pushes the protons to synchronize and spin together. This is nothing but a resonance. The resonance in the NMR. If you sum up all the vectors, the magnitudinal orientations, thus the resultant magnetic flux, and this flux in the form of electromotive force, this can be easily detected by a coil or an antenna. Electricity creates a magnetic field, and the magnetic field creates electricity. By using wire or ammeter, we can easily monitor this electricity. Using the ammeter, we place a magnet through the coil and will generate an electrical current through this wire. When we put the magnet back, the current flows in opposite direction. If we spin the magnet, we can get the sinusoidal alternate current. After RF pulse have been removed, the protons relax back and the positive charges repel each other, thereby creating a transverse magnetization, that is nothing but a T2 or spin-spin relaxation. There is no net energy transfer, immediately followed by T1 relaxation. That means the high energy protons release their energy into the surroundings, through which it falls back to the low energy state. There is the energy transfer. If we quickly put into another 90 degrees RF pulse, radio frequency pulse, the fully recovered protons produce a large transverse magnetization and gives a strong signal that would be recorded by a recorder. We can record, and if we listen back the echo shortly after the second radio frequency pulse has been dropped, the protons are still in high energy state with little regrowth of longitudinal magnetization. Immediately after supplying the second RF pulse, it pushes more low energy protons into high energy state and can produce a small transverse magnetization. The net longitudinal magnetization oriented 180 degrees are opposite to the main magnetic field. This configuration in protons will give a low amplitude or low energy wave. When we quickly listen to the echo, we can return to the signal and record the signal. You can see the typical instrument, NMR instrument, contains a source of radio frequency radiation, a receiver coil, a magnetic field that should be generated by the magnets, a sweep generated for varying the magnetic field, and a data acquisition system which comprises of a recorder and detector. Because the sample is placed between the poles of the powerful magnet and the superconductor. The radio frequency is generated in the coil connected to the RF oscillator. The detector coil is placed at the right angles in both the direction of the magnetic field and in the transmitter coil. The magnet is provided with the sweep coils which are used to vary the magnetic field. In general, the radio frequency is kept fixed. The magnetic field is varied until the resonance condition is reached. And here in NMR we can record the chemical shift, that is the shielding and de-shielding effects. When an atom is placed in a magnetic field, its electrons circulate about the direction of applied magnetic field. This circulation causes a small magnetic field at the nucleus which opposes the external applied magnetic field. This is called shielding effect. This ultimately decreases the effective magnetic field felt by the proton and shifting the signal to the higher magnetic field. This is called local diamagnetic shielding.